Hi everyone! Today we're going to talk about osmosis, and we're going to talk about osmosis by demonstrating osmosis in action at home. And to be perfectly clear, I mean in my home. So we are going to do a lab in my kitchen today involving potatoes. What we're going to do is we're going to cut up some potatoes into very similar sized pieces. We're going to soak those pieces in a variety of sugar solutions, and we're going to keep track of whether or not those potato pieces gain mass or lose mass over the time period that they're soaking in the sugar solution. That's the gist of what we're doing today. Now let's talk about a little background. Osmosis, as we know, is the movement of water through a semi-permeable membrane. Let's say this is a cell. Every single cell in our body is surrounded by a cell membrane, which is semi-permeable. The inside of that cell has some sort of solution in it, a certain solute concentration, and what we say is a certain water potential. Now, outside is also a solution. That solution on the outside of the cell may not have the same solute concentration as the inside of the cell. Now, osmosis is a movement from an area of high water potential to an area of low water potential. What that means is water will, on its own, move through a semi-permeable membrane from an area where there's lots of free water molecules compared to solute particles hanging around, into a region where there are fewer free water molecules and more solute particles hanging around. The cell doesn't have to expend any energy in doing this because the membrane is permeable to water molecules, but not these solute molecules. The cells that we're going to focus on today are, of course, the plant cells inside potatoes. If we put a potato cell, or a whole bunch of potato cells, inside a sugar solution, water will move from an area where there is a higher water potential into an area where there is a lower water potential, and vice versa. That will result in a change of mass of the potatoes if the potato cells have a different concentration to the concentration outside of the potato cell. Now for a little bit of terminology. The first term that we're going to need to know is hypotonic. If the potato cells are in a hypotonic solution, that means there's a higher concentration of water molecules outside the cell compared to inside of the cell. Now, due to the random motion of water, the water molecules will tend to enter the cell more than they will tend to leave it. What that means is that potato cell will gain mass, and we should be able to record that in our experiment. The next definition is hypertonic. If the potato cell is in a hypertonic solution, that means there's a lower water potential outside the cell compared to inside the cell. Water molecules will move randomly across that semi-permeable membrane, but what that means is the water molecules will tend to leave the cell more than they'll tend to enter the cell, resulting in the cell shrinking and the mass will decrease. The last term is called isotonic. An isotonic solution has the same concentration as the inside of the cell, which means water molecules will pass through that membrane freely, but the same amount will leave and the same amount will enter. So we should notice no change in mass of the potato cells in that case. Now onto the procedure. We're going to be cutting up a whole bunch of potatoes. We are going to use a cork borer to cut them up. That's going to cut them up into nice uniform cylinders. Then I'm going to chop up those cylinders into equal sizes. I'm going to group these potato pieces, 5 pieces per sugar solution. Why 5? Because I'm not going to be perfect at cutting up these tiny little potato cylinders. So I want to make sure I've evened out the size and shape as much as possible. The other thing we're going to do is we're going to weigh the potato pieces before and after they've soaked in the solution. As you can see, I've already placed saran wrap on all of these different sugar solutions to prevent evaporation. The sugar solutions I'm using are 0 moles per liter, which means this is just tap water. Yes, I'm using tap water because remember, this is an experiment at home. 0.1 moles per liter of sugar solution, 0.2 molar, 0.3 molar, 0.4 molar, 0.5 molar, and 0.6 molar sugar solution. Now, as I'm doing the procedure, what I want you to think of are some sources of error that could come into play in this particular lab. Yes, I'm trying my best and I'm being as careful as I possibly can. However, this is an experiment at home. So I want you to think and write down any things that occur or any parts of this procedure that you think could affect our final result. That would constitute a source of error. So now that I've got my potatoes, let's go ahead and get started. Okay, so we've got our point of view camera set up and we're ready to cut up some potato. I'm going to need to use this cork borer to cut up the potatoes in nice cylindrical slices. I've also got this really old ruler with very hard to see tick marks, and I'm going to measure 2 centimeter cuts on the cylinders that I cut out. As I'm cutting out each potato piece, I'm going to push it out with my glass rod. So here's potato cylinder number 1. I'm going to need to cut off the ends because, as you can see, there's some potato skin, and I don't think the water is going to pass very nicely through that potato skin. I want all of the outside of my potato pieces to look nice and uniform. So I'm just going to start by poking a whole bunch of holes in this potato. Now take a look at what happened with this one. I was a little too close to a previous cylinder that I had bored in my potato, and so this one has a piece cut out of it. So I'm not going to use that piece. I'm going to omit any pieces that I find where I actually messed up cork boring my cylinder. So now that I've got some pieces cut out to start, and yes, I probably will need a lot more, I'm going to show you the process of chopping them up into nice, uniform sized cylinders. So like I said before, I want to chop off the end, which is the skin. I'll put that in my garbage pile off to the side. Now these cylinders, I would like them to be 2 centimeters long. So I'm going to measure this out on my ruler, and I'm going to make a chop as vertically as I can at 2 centimeters. So that's about what I want all my potato pieces to look like. So I'm going to go ahead and chop these up into 2 centimeter pieces and make a pile off to the side. Now, one thing you may be noticing that I'm noticing, when I look at the cross section of these potatoes, they're not all a uniform color. So there are some inconsistencies on the inside of this potato. Maybe that will affect my results, maybe not. Might be a good thing to make a note of. So right now I have 15 potato cylinders cut. I'm going to need 5 cylinders for each one of my solutions. So I've got a ways to go. I'm going to skip ahead a little bit for you guys. Okay, so here we are. I finished cutting up all of the different cylinders. Good news is I've got all my cylinders from the same potato. So that's another variable I didn't realize I'd be able to control, but I was. Now we're at the part where we have to measure the mass of each of our groupings of 5 potatoes. Once I measure the mass of each grouping of 5, I'm going to place them on one of my cards so I remember which grouping goes with which solution. So I'm going to start by turning on my scale. And I'm just adjusting my lighting slightly so there's no glare on the scale so you can clearly see every reading. My scale is zeroed already, which is fantastic, so I'm going to add my first 5 pieces. Now, as you're following along at home, I want you to record these masses for yourself. Now I'm going to place those potato pieces right here, ready to go for later on. Next grouping of 5. My scale is still zeroed. Next group of 5. So notice that even though I was very careful in cutting, each of the masses of my groups of potatoes are a little bit different. But that's okay, we'll take that into account in our analysis. So now what I need to do is I need to add the potatoes to my sugar solution. I've got the mass, I'm ready to add the potatoes. I need to use my phone as a timer now. So as soon as I add the first grouping of potatoes, I'm going to click Start on my timer. I'm going to add the 0 molar solution first. Now what I'm going to do is I'm going to stagger when I add the potatoes to each subsequent solution, because I want every single grouping of potatoes to be soaking for exactly the same amount of time. So I want to give myself time at the end of my experiment so that I have time to dump the potatoes, pat them dry, and then measure the final mass before I have to do the same thing to the next solution of potatoes. So I'm going to stagger all of my potatoes by exactly 1 minute. So I'm going to keep an eye on the timer. You can watch and see how closely I am to keeping up with my timer. And I'm going to stagger when I add the potatoes to every single solution. Okay, now they're in and we wait. I'm gonna wait about 30 minutes until I start taking the potatoes out of our first solution. And I'm also going to give the potatoes a stir once or twice during those 30 minutes. So I'm just about ready to take the potatoes out. It's been about half an hour, and I just wanted to show you this setup. I've got all my glasses ready. What I'm gonna do is dump every glass into this strainer, block the potatoes on this paper towel to remove any excess water, but I'm gonna be careful not to squeeze them. I'm gonna immediately weigh them, record the mass, well, you're gonna record the mass, and then I'm gonna do the same thing to the next glass, the next solution, but I'm gonna stagger it by 1 minute. So I'm just about ready to begin with my first solution. I'm gonna time this for exactly 30 minutes. And the mass is 3.84 grams. This one is 3.80 grams. Next one is 3.63 grams. This one is 3.71 grams. This one is 3.62 grams. This one has a little bit on it. Looks like I missed a bit of skin. I'm gonna leave that on as I measure the mass since it was part of the mass to begin with. This one is 3.59 grams. This one is 3.63 grams. Now we're all done collecting our data. Now it's up to you to do some analysis. Now you may notice that our final masses varied quite a bit, and they may be different compared to our initial masses, but remember, the potato groupings didn't all start at the same initial mass. So a better thing to compare than just the final masses of each set of potatoes is to figure out how much the potato masses changed as a percentage of their initial mass. Now that you've tabulated your data, you're going to use a spreadsheet like Excel or Google Sheets to plot your results. Your job is to figure out what is the osmotic concentration of the potato cells. Good luck!